Dear colleagues, let me start the lecture about the nervous system. You will study the nervous system in anatomy very much. You already started with peripheral nervous system and you will go on in central nervous system. So you will know perfectly these two parts of the nervous system. Here in histology, we will deal with only several parts of the nervous system. Concerning central nervous system, we'll look at the structure of spinal cord, we'll look at the structure of the cerebellum and cerebrum, that means terminal part of a brain, so-called telencephalon, and at the end of my lecture there will be something about nerves and ganglia. Please don't expect that I'll say everything about the histological structure of nervous system during one lecture. It is very complicated. You will see some histology also together with anatomical description of the nervous system. And if somebody of you will want to study the nervous system in detail, can look at many sources, many textbooks of neuroanatomy, neurohistology. This is a big science and it isn't possible to say everything during one lecture in the first year of medicine. So I will start with central nervous system, but only with these three portions as I mentioned, and then there will be peripheral nervous system. The division of the nervous system into the central and peripheral isn't the only possibility how to classify the portions of the nervous system. Nervous system can be also divided into somatic and autonomic. Somatic nervous system is this part of the nervous system which is working with voluntary motility and sensitivity. So that's why we divide it into somatosensitive and somatomotoric system. The second is autonomic or also vegetative nervous system, which is sympathetic and parasympathetic. This division is based not only on the position of nuclei. You hopefully know from anatomy that nuclei of parasympathetic nervous system are situated in the brainstem and in the sacral segments of the spinal cord, and on the other hand, sympathetic nervous system is starting by its preganglionic neurons in thoracic and lumbar area of the spinal cord. But there is also a difference in the neurotransmitter. Peripheral neurotransmitter of parasympathetic system is acetylcholine. Peripheral neurotransmitter of sympathetic system is norepinephrine or noradrenaline. So that's the second system, autonomic nervous system. Also, we'll look at some parts of it in my lecture. The nervous system is composed almost only from the nerve tissue. Not completely. There are also vessels. There are also meninges, which are composed of several types of connective tissues, and all these structures belong to nervous system, but the nerve tissue is really the main tissue of the nervous system. And as it is some time we discussed the nerve tissue, and I think that it is good to repeat sometimes the basic information. I will show several pictures concerning nerve tissue at the beginning of my lecture. Hopefully you remember that the nerve tissue is composed of many cells and only small, almost negligible volume, negligible amount of the extracellular matrix. These cells are typical by the processes having on their surface. Usually these processes are quite long, and the cells of nerve tissue are of two types or two families. The main of them are neurons. The second, but also important and more numerous, are glial cells or cells of neuroglia. If you look at a picture, which is just illustration, you can see several neurons, which is possible to recognize according to their typical very light nucleus and central nucleolus. The number of the neurons in this picture isn't high. 1, 2, 3, 4, 5, 6, 7, maybe 7 neurons. And all other nuclei here belong to glial cells. So you can see that they are more numerous, but usually smaller. Not every time. We will see a part of a brain where neurons are smaller than glial cells, but usually it is valid information. The nerve tissue can be studied by classic histological techniques, by the synoptic staining, hematoxin, eosin, or other synoptic stainings, but in these stainings we can never see all details of the nerve tissue. Mostly we don't see thin and frequently very long processes of neurons and glial cells. If we want to see more, we should use special neurohistological methods, special neurohistological stainings. Probably the most modern are various immunocytochemical or immunohistochemical methods. In this picture upstairs there is a fluorescent labeling of the antibodies, and we can see very well the shape of a neuron and their contacts. But there exist also classic neurohistochemical methods, like for example Nissl methods, which is based on the toluidine blue, staining the Nissl substance, and also nucleolus is very well stained. We will use one slide in our practical training, so you will see the Nissl method of staining. Another method is Weigert's method or Luxol blue, which isn't here visible, which are staining myelin sheath. You know hopefully that myelin sheath normally are poorly stained. They are very light because the myelin is almost destroyed during the classic histological procedure, but there exist special methods which preserve the myelin sheath, and we can see them around the axons, for example here in the peripheral nerve. And very good and very frequently used are impregnation methods, where usually silver is used as the metal for this impregnation. There are several types of silver impregnations. In this case, neurofibrils, which means bundles of neurofilaments, are visualized in neurons, mostly in their dendrites, and we can see not only bodies of neurons like this or this, but also their long and branching processes. So that's possible to use, and impregnation is also a part of our set. A neuron is a cell which hasn't got only the typical nucleus, which I already described, but also another components. Nucleus and most organelles are situated in the neuronal body, which is also called soma or perikaryon. Instead of nucleus, there are several fields containing ribosomes and rough endoplasmic reticulum, which stained by Nissl staining or another basic dyes like hematoxylin, and which represent Nissl bodies known in light microscopy. There is also quite well developed Golgi complex, usually divided into several fields, similarly as a Nissl substance. You know that Golgi complex is cooperating with rough endoplasmic reticulum, so it isn't surprising that each Nissl body has got its own Golgi complex for the processing of the products. Many mitochondria are in a neuron. Normal pair of centrioles, which is here situated and usually near the nucleus, and which is important for the production of microtubules. Microtubules represent the main elements of cytoskeleton of a neuron. They are transported after development near the centrioles to the processes, to dendrites or to axon, and they are important for the transport of many substances, and mostly in the axon they are important for the transport of neurotransmitters in their synaptic vesicles. I hope that it can be enough. You already heard it in another lecture, so let's look at several pictures only. Neuronal body in the light and electron microscope. These pictures well correspond. Nucleus is very light, rich in euchromatin in the light microscope, and of course the same situation is here in the electron microscope. Almost only euchromatin is here and big nucleolus. Cytoplasm in the light microscope is rich in Nissl bodies, basophilic grains, where we know that there are many ribosomes and rough endoplasmic reticulum, and really in the electron microscope we can see huge accumulations of these organelles together with the Golgi complex. Neuroglia is a complex of cells which are situated in central and peripheral nervous system, and thus they are classified according to this location into central and peripheral glial cells. Central glial cells are more numerous. We recognize 4 categories of central glial cells: ependymal cells, which are lining cavities of central nervous system; astrocytes, which can be protoplasmic in gray matter and fibrous in white matter; oligodendrocytes, which are responsible for the axon myelination in the central nervous system; and microglial cells, which are very special. They have another origin. They originate in the monocytes, or their precursors are monocytes. They belong to mononuclear phagocytic system together with macrophages, osteoclasts, dendritic cells, and they are responsible for the cleaning of the nervous system, for the protection of the nervous system, for the phagocytosis of some damaged components of the nervous system. Peripheral nervous system contains only 2 types of cells: Schwann cells and satellite cells. Schwann cells are responsible for axon myelination in PNS, and satellite cells accompany neuronal bodies in ganglia. I mentioned when I described types of astrocytes are mentioned gray and white matter. Gray and white matter are 2 components of the central nervous system. I hope you know them, not only from anatomy but also from high schools. It is common knowledge. Gray matter is this part of the central nervous system where you can find bodies of neurons. So here impregnation was used, so bodies are these big black structures. Bodies of neurons are situated in gray matter, and we can recognize gray matter according to the presence of these bodies. But of course the bodies of neurons isn't the only thing which we can find there. There are also their processes, their dendrites, and also the proximal portion of their axons. There are also glial cells, already mentioned protoplasmic astrocytes, but also oligodendrocytes which myelinate the proximal parts of axons, or which myelinate axons of another neurons coming here from surrounding tissue. Microglial cells should be here. So the gray matter is quite rich in structures, and we use a special word neuropil for the mixture, let's say mixture of processes which are found in gray matter. These can be processes of neurons, dendrites and axons, but these can be also processes of all glial cells. Astrocytes have got long processes, microglial cells have got long and branching, especially branching processes. Oligodendrocytes have got short processes, but also some processes are there, and all this material which we can see here and in which we are not able to clearly distinguish which process belongs to which cell, we call this material neuropil. On the other hand, white matter, which is at the right side of my picture, lacks bodies of neurons. It is the main thing according to which you can recognize it. Concerning neurons, it contains only axons, maybe some long dendrites as well, but usually axons only. These axons are frequently myelinated. We can see them here. These rings in a white matter having dark dot in the center. These are myelinated axons, or axons with myelin sheath, but some axons here are also unmyelinated. Then there are also fibrous astrocytes, very many oligodendrocytes which are responsible for this myelination. Microglial cells are in the whole CNS. So all this material is here found, and this is the composition of white matter. We don't use the word neuropil for the mixture of processes in a white matter. This mixture of processes is not so complicated. Axons are usually myelinated, so recognizable, so we don't use neuropil here. Of course, all matters, or both matters of CNS, are vascularized. The capillaries can enter white or gray matter, and they are surrounded by a special sheath composed of glial cell processes. So capillaries in the tissue of CNS are not accompanied with connective tissue, but bigger vessels. Yes, bigger vessels are accompanied with connective tissue, which is a invagination, or which is a derivative of the pia mater, one of meninges. Gray matter in another picture without impregnation, hematoxin and eosin, so we can see only proximal portions of processes, here dendrites, processes of this neuron. We can see some nuclei where we can recognize smaller spherical nucleus of oligodendrocytes and bigger ovoid nucleus of astrocytes. If there is a microglial cell, its nucleus should be long and very dark. It isn't, unfortunately, in this picture. And let's look at this material. It is the already mentioned neuropil, mixture of unrecognizable processes. Picture from a gray matter showing a neuronal body and its nucleus, which is here, and then neuropil again, where are many processes which are not well recognizable. Only the bigger ones can be recognized as non-myelinated or myelinated axons. Myelinated axons are well recognizable because they have got this dark, in electron microscopy, very dark, very electron dense myelin sheath. You know that myelin sheath is composed of several layers of a membrane which is electron dense, and so the picture isn't surprising here. And it is a picture of neuropil. Many processes, some of them are probably dendrites because they are branching and they are quite thick. But what about all these small things here? They are usually processes of glial cells. Let's not forget to synapses. Mostly in gray matter, there are many synapses between axon dendrites or axon and bodies of neurons, or there can be also axonal synapses. Look at the part about the synapses in your textbook. Please repeat how do they work, how the neurotransmitter, which is situated in the synaptic vesicle, is released on the presynaptic membrane, transferring the synaptic cleft bound to the postsynaptic membrane, and the receptors here in the postsynaptic membrane are changing the polarization of the membrane, and the signal can be then transported along the dendrite or along the membrane of the body to another places. White matter doesn't contain neurons, or it isn't well said, but doesn't contain neuronal bodies, pericardia, or neurocytes also it's possible to say. So they are not situated in the white matter. So if you see there any nucleus, it must be nucleus of a glial cell, and it is really possible to recognize bigger and frequently ovoid or irregular astrocyte nuclei, spherical smaller oligodendrocyte nuclei, and elongated, thin and highly condensed nuclei of microglial cells. And I think that the repetition of the nerve tissue can be finished at this point, and let's look at the microscopic anatomy of some parts, some selected parts of the nervous system, and let's start in spinal cord. Spinal cord, medulla spinalis, is an organ which has got white matter on the peripheral part, or in the peripheral part, and gray matter in the center. If you look at the section of a spinal cord, or to the microscopic slide specimen of the spinal cord, you will quickly recognize its gray and white matter. Gray matter has got very specific shape in the section. Somebody says that it is like a butterfly. Somebody says that it's like a letter H. Butterfly is nicer than letter H, so I prefer the first meaning. Nevertheless, this shape is variable in various segments of the spinal cord, but usually it's like this. This gray matter butterfly has got ventral and dorsal horns, and sometimes, mostly in the thoracic region, also lateral horns. Ventral horns are wider, thicker, and don't reach the surface. There is a piece of white matter between the tip of the ventral horn and the surface of the spinal cord. Dorsal horns are thinner and they reach the surface of the spinal cord and divide the dorsal white matter into two funicles. Gray matter is situated also in the central area of the spinal cord, around the central canal. Central canal belongs to the hollow spaces, or to the system of spaces in the central nervous system, together with ventricles. We will see in the microscopic sections that it is lined by ependymal cells, which will resemble simple columnar epithelium, and the gray matter around the central canal forms dorsal and ventral commissure. There are gray commissures, and that is a description of the gray matter of the spinal cord. You use word horns, or in Latin cornua, if you speak about the two-dimensional section of the spinal cord. If you speak about the three-dimensional structure of the spinal cord, let's prefer a term column. So there are dorsal columns, ventral columns, and lateral columns, which are visible in the section as horns. White matter is divided into three funicles, of course symmetrically in both halves of the spinal cord. There is a dorsal funicle, lateral funicle, and ventral funicle. Ventral and lateral funicles are connected around the ventral horn. Dorsal and lateral funicles are usually separated by dorsal horns. There exists also a deep fissure situated in the ventral surface and allowing the vascularization of the gray matter. Large vessels, or large, quite big vessels, are entering this fissure and come to the vicinity of the central area of the spinal cord. On the other side, on the dorsal side, there is just sulcus, which is quite shallow, and then glial septum separating two dorsal funicles. On the other hand, ventral funicles are connected by a white commissure here. So white commissure is only one. Gray commissures are two, dorsal and ventral, around the central canal. I think that that's still almost anatomical description. Let's go to histology. Let's finish this anatomical description by a picture of a whole spinal cord. It isn't human spinal cord. It is a spinal cord of mouse, so its shape is a little different than you know from anatomical textbooks, but we can see all the important structures. Impregnation was used, so the gray matter is very dark, and we can see ventral horns, dorsal horns, which have another shape in mouse than in human, central gray matter having two commissures, ventrally is deep fissure and wide commissures, and there are ventral funicles, lateral funicles of a gray matter, dorsal funicles, which are separated by a glial septum. Roots of spinal nerves are here. Dorsal roots are well visible. It is a bundle of several bundles of axons are entering the spinal cord via the dorsal roots. They go to the dorsal horns. And now finally human spinal cord and little higher magnification, so we can see central canal, which is well visible in all slides and which you will use for orientation. You'll see the ependyme resembling a simple columnar epithelium. In this staining and in this magnification I wouldn't recognize whether it is epithelium or not. It is possible to recognize only using special staining or electron microscopy, when you will find that there isn't any basement membrane or basal lamina, that these cells, these ependymal cells, are integral components of the nerve tissue. Here we can see gray matter around the spinal cord, forming the wings of our butterfly, and as we here can see fissure, ventral median fissure, we can be sure that here is ventral surface or ventral side of the spinal cord. But if we don't see a fissure, we should recognize the ventral horns according to presence of big neurons. Ventral horns contain much bigger bodies of neurons than dorsal horns. Biggest neuron in dorsal horn is, for example, this one, and you can see the difference. And yes, here is the septum, dorsal medial septum, two dorsal funicles, two ventral funicles, which are not connected here, but should be by a wide commissure. Yes, okay. So I think that the rough description of the spinal cord can be finished, and now let's look at the types of neurons which are found here. The spinal cord contains two families of neurons. It contains efferent neurons, which also can be called root cells, and interneurons. Efferent neurons and interneurons. Maybe you are surprised why not also afferent neurons, but I explain it later. They are really not situated in the spinal cord but in spinal ganglia, and they send only their axons, only the central branches of axons to the spinal cord. Efferent neurons can be motor or autonomic. Motor neurons are much bigger, and they are situated in ventral horns. Motor neurons, or bodies of motor neurons, are situated in ventral horns. Autonomic neurons are smaller, and their bodies are situated mostly in the central central gray matter, and also if present in lateral horns. Then we have got interneurons. Interneurons are neurons which are connecting all other neurons and which don't send so long axons to leave the central nervous system. It is important. They can contain long axons, but their axons can never leave the CNS. They connect only neurons situated in the CNS. In the case of the spinal cord, there are two groups of interneurons. The first of them are so-called connecting neurons, and the second are funicular neurons. Connecting neurons, as you can read here, connect neurons in the same or neighboring segment. So they have got only short axons, they belong to Golgi type 2 neurons, they connect neurons in the same or neighboring segment, and they can be intercalated, commissural or associative. I explain it in the next schema. Funicular neurons belong to Golgi type 1. They have longer axons which leave a gray matter, go to white matter, and can go either cranially or caudally and connect more distant segments, or they can connect the segment of a spinal cord with some part of a brain. So they represent the typical neurons of the spinal tracts. This schema can explain differences between the three types of connecting interneurons. Connecting interneurons can be, as I already said, intercalated, commissural, or associative. Intercalated neurons are smallest. Their bodies are usually situated in dorsal horns, and they connect afferent axon coming from the dorsal root with the efferent, usually motor neuron situated in the ventral horn. Their axon doesn't leave gray matter, and even more, their axon doesn't leave 1/2 of a gray matter. So this is intercalated neuron. Commissural neurons are similar, but their axon is going through the gray commissure to the other side. So it connects afferent axon with the motor neuron contralaterally. Associative neurons, associative interneurons, it is maybe better visible here. They are also situated in a gray matter. They can be either in dorsal or in ventral or horn or in central area, and they send axon which is going either to the cranial or caudal neighboring segment, but not longer. It isn't longer, and it usually doesn't leave gray matter. It is still in the gray matter and connects some afferent endings with the motor neurons in the neighboring segments of the spinal cord. So these were three connecting neurons, connecting interneurons. Funicular interneurons are bigger. Their bodies, for example, this is the interneurons. Their bodies are in a gray matter, but they send the axon to the white matter, and it can go usually upstairs. It can go usually to brain or to some cranial segments, and they are components of funicles, of course. The biggest and most typical neurons for the spinal cord are motor neurons. They are situated really only in the ventral horns, and you recognize them according to high accumulation of Nissl bodies, large spherical nucleus. When is a section through the center, there is also nucleolus. And motor neurons are typical multipolar neurons having more dendrites and one axon. It is sometimes possible to recognize the processes. For example, here is a process having Nissl bodies inside, so it is a dendrite. Probably here is a process which doesn't contain Nissl bodies inside, so it is probably axon. If you want to be sure, you should use special immunohistochemical staining of microtubule associated proteins, but you can guess that it is the axon, it is the dendrite. Here another axon is usually probably, or here some proximal portions of another dendrites are visible. In between the bodies of the motor neurons there are many glial cell nuclei and neuropil, as was already said. It is a detail of a motor neuron. Axon, which is not branching and which lacks Nissl bodies. It should contain only microtubules, some mitochondria, maybe some smooth endoplasmic reticulum, but no Nissl bodies, no ribosomes. And here is a dendrite. We don't see its branching, but here we can see branching of a dendrite and the content of Nissl substance inside. Of course, after impregnation we can see more. Multipolar neurons are here visible. And now we are in a white matter, where are usually myelinated axons. Myelinated axons of spinal cord are parallelly oriented, parallelly organized. So if you have a good section going really transversally, you can see axons as eosinophilic dots, which should be normally in the center of each myelin sheath, but as the myelin is extracted here, the axons tend to be situated peripherally. So we can see axons, and there we don't see myelin sheath. Here we can see spaces where myelin sheaths should be, if not extracted. Yeah, so myelinated axons are here. Of course, many unmyelinated axons and another processes. And if you see a nucleus, it must be a nucleus of a glial cell. And in this area, usually oligodendrocyte. Maybe this biggest one is a fibrous astrocyte. It isn't oligodendrocyte. It's irregular and quite huge, but the other small spherical nuclei, frequently with visible nucleolus, they belong to oligodendrocytes. Oligodendrocytes can myelinate more axons because they have processes, and each process can myelinate one. So the number of oligodendrocytes can be smaller than the number of myelinated axons here. It is a detail of a central canal. And really, we don't see any basement membrane under the under the eppendyme, so it isn't probably proof. Real proof should be immunohistochemistry of collagen type 4, for example, or laminin. If you don't find it here, you can really say there isn't any basal lamina. Basal lamina isn't visible in a light microscope. Nevertheless, there is the irregular border between the eppendyme and remaining nerve tissue, so we can guess that really there is no basement membrane and that the cells of the eppendyme are only epithelium-like or epithelioid, and they are integral components of nerve tissue. There is simple epithelium on their apical surfaces. They sometimes can contain cilia, or they every times have microvilli on the surface. Cilia are responsible for the movement of cerebrospinal fluid in the canal, and microvilli are responsible for some absorption of fluid from the cerebrospinal fluid. Here is a picture of the apical surface of the eppendyme in the scanning electron microscope, and you can see that some ependymal cells have got small tufts of cilia, but another ones haven't got them and are equipped only with short microvilli on the surface. Yeah, okay, so we can leave the spinal cord and you will see all these structures in practical training. And let's go to cerebellum. Cerebellum has got gray matter in two locations. Gray matter of cerebellum isn't only centrally, where it forms cerebellar nuclei, but cerebellum is one of two parts of a brain where gray matter is also on the surface, forming a cortex. So cerebellum and also the telencephalon have got cortex. Cortex of cerebellum is composed of 3 layers. You can read them here and we will speak about them in details. White matter is situated between the cortex and cerebellar nuclei, and it has very special appearance. It is like branches of a tree. You will see the cerebellar section in the anatomical department. I am sure they will show it in macroscopic specimen, and you will see a white matter forming so-called arbor vitae. Arbor vitae, tree of life, is historic name of the special shape of white matter in the cerebellum. Let's look at the cortex at first. Cerebellar cortex has got 3 layers, which I already mentioned. Their Latin names are here: stratum moleculare, gangliosum and granulosum. If you use English names, you can speak about molecular layer, ganglionic or also Purkinje cells layer, because Purkinje cells are the main cells of this layer. And the third is granular layer, so molecular, Purkinje cell layer and granular layer. I will start from Purkinje cells. Purkinje cells, although situated in the middle layer in the stratum gangliosum, are so big that they in fact occupy all 3 layers. They have dendrites, which are oriented peripherally, and so they are situated in the molecular layer. The dendrites of Purkinje cells are highly branched, and their branching is in one plane, so if well sectioned we can see branching dendrites in the light microscopic picture. Even without impregnation you will see them in our slide. Bodies are in one line, one layer in the middle of the cerebral cortex, and they are forming stratum gangliosum, and axons of Purkinje cells go down, go centrally, go through the granular layer to white matter and to cerebral nuclei. So Purkinje cells are really the main cells here. You already saw them. We demonstrated neurons and size of cells in the specimen of the cerebellum. So you already know Purkinje cells, and you will see them once more in our practical training. Their dendrites are in molecular layer and occupy the main volume of the molecular layer. So the molecular layer doesn't contain many cells. There are two types of small neurons. Stellate cells are in a molecular layer, which are only small associative neurons having short axons, don't leaving the molecular layer. And then there are also basket cells, which have a little longer axons, which form synapses on the surface of the Purkinje cell body. Of course, there are also glial cells in the molecular layer, and some axons of another neurons, like small granular neurons from the molecular layer, or also there are so-called climbing fibers coming from deeper areas of a cerebellum, which can also enter the cerebellar cortex. So molecular layer of the cerebellar cortex is very rich in processes, very rich in dendrites or axons, and the number of cell bodies there is quite small. And then we can go through the layer of Purkinje cells to granular layer, where are really very many neurons. You will see it in slide. Two neurons are here, small granular neurons. They are very numerous. They are so small that they are in fact one of smallest cells of the body. They measure only 4-5 micrometers, so less than erythrocyte and less than glial cells. So small granular neurons in the cerebellar cortex are really very small, but they have got axon. Their axon is going through the layer of Purkinje cells and is branching as a letter T in the molecular layer, forming so-called parallel fibers, anastomosing or forming synaptic contacts with dendrites of Purkinje cells. Small granular neurons are very numerous, you will see it. Much less numerous are large granular neurons, which are also called Golgi neurons. They measure about 50 micrometers, so they are much bigger than small granular neurons, but still smaller than Purkinje cells, which can measure more than 100 micrometers. These large granular or Golgi neurons have an opposite orientation. Their dendrites go up to molecular layer here, but axons are branching near the body coming to synaptic contact with small granular neurons and don't leave the granular layer of the cerebellum. The last thing which I still didn't mention are so-called mossy fibers. I was spoken about the climbing fibers which come from deeper areas of the cerebellum and go to Purkinje cell or molecular layer. Mossy fibers are shorter, they go only to granular layer and form synapses with axons of small with dendrites of course, of small granular neurons. So you can see the complex, very complex connection of all components of cerebellar cortex and also white matter of cerebellum. And now let's look at several pictures. So this is the cortex of cerebellum. Here is the surface, normally covered by meninges, but in this case free. And the first layer, which can be called molecular layer, or if you prefer Latin, you should say stratum moleculare. Stratum moleculare. And moleculare, or also stratum cinereum. And you can see that there are many processes. The neuropil is here huge, but the cell bodies are only rare. In this magnification we aren't able to recognize the cell bodies here, but some of them should be stellate or basket neurons, and other ones can be astrocytes, oligodendrocytes, microglial cells. Of course, capillaries are here as well. For example, here up I see two nice sections, some nice longitudinal sections of capillaries containing several erythrocytes. Yeah, so don't forget, or never forget, that the CNS is well vascularized, that there isn't any layer of connective tissue between capillary and nerve tissue, and so the capillaries become integral component of the nerve tissue here. Then we can find a layer, a row of big cells, big neurons having a typical shape like pears. For example, here is a pear shape of a cell, piriform shape. And we know that the Purkinje cells have got their dendrites on the surface oriented to the molecular layer. So this or this or this process, here is very long section, or here, you look at this long and branching process. These are dendrites of Purkinje cells. They are well visible, they are better visible than axons because they can contain Nissl bodies or some ribosomes, so they are basophilic and so well visible in the otherwise eosinophilic neuropil. On the other hand, axons of Purkinje cells are hard recognizable. Maybe here I can see some line going from the base of the Purkinje cell to the granular layer, but usually we don't find it. Maybe here as well. So axons of Purkinje cells go to this granular layer, and it is here. So it is granular layer or stratum granulosum, and look at many cells here. Most cells are accumulating in the groups. These groups, composed of small granular neurons, are called cerebellar glomeruli. Typical cerebellar glomerulus is, for example, this group of small granular neuron having eosinophilic central cord, central field, where mostly synapses with mossy fibers have place. Another cerebellar glomerulus is this, with the central cord, or where can I see such typical here not, but it will be better in higher magnification. So the small granular neurons tend to accumulate in groups, so-called cerebellar glomeruli. Cerebellar glomeruli, having nothing common with renal glomeruli, of course. Only the shape, the spherical shape, probably resembled the renal glomerulus to somebody, and so the name is like is glomeruli. Okay. So this is the small magnification of the cerebellar cortex, and here will be several pictures in higher magnification. These two pictures are focused on the Purkinje cell and branching of its dendrite. Of course, if you have just hematoxin, eosin, you don't see so much. You can see dendrites. Usually there are two main dendrites going from the Purkinje cell. Two main dendrites. And if you use impregnation, you can trace their branching to much thinner branches. It's the advantage of these dendrites is that their branches in one plane usually. So if you have perfect section, you can see whole branching, whole system of dendritic processes. And here is, of course, stratum moleculare, here is stratum granulosum. Another picture showing big system of dendritic branches of Purkinje cell. And if you use electron microscopy, you can see that there are so-called spines on the dendrites, so the superficial area of the dendrites here in Purkinje cells is very big, and there can be many thousands synapses between axons. There can be axons of granular cells, there can be climbing fibers, and this dendrite. Yeah, this is the promised picture showing cerebellar glomeruli a little better. Yeah, let's look at the granular layer showing spherical groups of small neurons having eosinophilic central fields. These central fields contain dendrites of small granular neurons and mossy fibers, which are coming from white matter and form the synaptic contacts with these dendrites here. Occasionally you can find bigger neuronal body in the stratum granulosum, but really not many Golgi neurons are here. But what we can see in this higher magnification are several nuclei bigger than nuclei of small granular neurons. And when you know the size of small granular neurons, you aren't surprised that these larger nuclei belong to astrocytes. Astrocytes are bigger cells than small granular neurons. And of course again some vessel here. No. Organization into the cerebellar glomeruli is visible here as well. Okay, so we can leave a cerebellum. We'll skip all other components of brainstem, mesencephalon, pons, medulla oblongata. We'll skip also diencephalon, which is its hypothalamus, thalamus, and we go directly to the telencephalon, to the cerebrum, and we'll focus on its cortex again. Only, in fact, we'll study only the cortex of the cerebrum. We'll not study basal ganglia, which represent central gray matter of the telencephalon. We haven't got them in our slides. Our slide is like this. It contains cortex, which is composed of gray matter, and white matter, so-called centrum, semi-ovale, central material of the cerebrum, which is composed of gray matter. White matter. How to recognize it? White matter is this. You don't see any big cell body in this small magnification. Nothing more can be visible, but there aren't any bigger bodies. There are some vessels and small cells. You can be sure that they belong to glial cell family. And here is a gray matter. You can recognize it according to two things. One of them is that it is on the surface. Here is the surface of the cortex covered by some connective tissue. And the second thing is that even in such small magnification, you can identify bigger cell bodies, isn't it? So, gray matter, white matter. Cerebral cortex is composed of several layers as well, like cerebellar cortex, but the recognition, the borders of layers here are not so clear, so it will be more theory than practice. In practice, you can distinguish usually only some of these layers clearly, and the others you can just guess where they should be. But you will see the areas where are less cells and more cells. You will see areas where the cells are small or where the cells are bigger. The biggest cells are in the fifth layer. They are so-called large pyramidal cells, which can measure more than 100 micrometers, like Purkinje cells of a cerebellum or motor neurons in the spinal cord. Also these cells, these large pyramidal cells, which can be called pyramids of beds. So these cells belong to the family of very large neurons. Here is a picture from the cortex showing several neuronal bodies. We don't know in which layer it is, so it is impossible to speak about the type of cell here. Among the neuronal bodies is neuropil, you already know it, and nuclei of glial cells. These are probably oligodendrocytes here. And vessels, of course. is a capillary with red blood cell, and here is a capillary with lymphocyte. It's interesting. Yeah, okay. Here is another capillary showing erythrocytes. And look at it, there isn't any connective tissue, there is just endothelium, nuclei of endothelial cells are here, and nervous tissue on the surface. We know that capillaries are covered by processes of astrocytes forming perivascular limiting membrane. You already know it from general histology. Which are the cortical neurons? Cortical neurons are classified as excitatory and inhibitory. It is one classification here. You can see that excitatory are more numerous. They represent 60 to 70% of neurons. Inhibitory are less numerous. How to distinguish excitatory and inhibitory neurons in morphology, in histology? You should look for the dendritic spines. If you find them, the neuron belongs to excitatory group. If the dendrites are smooth, that there aren't any dendritic spines, which can be visible in impregnation in the light microscopy. You don't need electron microscopy for this. You can be sure that the neuron is inhibitory. Another way how to recognize them is immunohistochemistry. You'll focus on the neurotransmitter. Excitatory neurons of cerebral cortex contain glutamate. Inhibitory neurons contain gamma aminobutyric acid, well known under the abbreviation GABA. So they are so-called GABAergic. The second classification of cortical neurons is efferent neurons and interneurons. It is very similar, as you know, from spinal cord. Efferent neurons are neurons sending their axons to some very distant areas. They form nerve tracks, and their shape is usually pyramidal. So the efferent neurons of the cortex are called pyramidal cells. Pyramidal cells, it is the typical name of excitatory neurons or efferent neurons of the cortex. So they we can see pyramidal. I wanted to draw a shape of the pyramidal cell, but the picture skipped. So we can see the typical shape of pyramidal cell. Of course, it is triangle in the section, and we know that the axon of the pyramidal cell is starting in the middle of the base of this pyramid, of the shorter side, and the main dendrites are going from the tips. So if you look at this impregnated neuron in the right picture, this is one main dendrite, second main dendrite, the third main dendrite, which go from the tips of the pyramid. Of course, here in the base can be more main dendrites. We can see just section, but here apically is really only one, and probably here we don't see exactly is the axon. It isn't so well stained in the impregnation because it doesn't contain so many neurofibrils. Okay, I'll go back to the previous picture because I want also to say something about the interneurons. Cortical interneurons are very many. They have short axons, sometimes it's very interesting branching, these axons don't leave the cerebral cortex, and some of these neurons are excitatory. They are called spiny stellate cells, but many other are inhibitory. Inhibitory neurons of the cortex are classified according to synaptic places. These which have their synapses on bodies or proximal dendrites that means which send axons forming synapses with bodies or proximal dendrites of another neurons. They are called basket cells. You know basket cells from cerebellum, they are very similar. These inhibitory interneurons which send axons forming synapses on distal dendrites of another neurons form next big group, and there are also cells with axonic synapses. Their axons are very specifically branched, forming something like chandeliers, so that's why they are called chandelier cells. I think it isn't necessary to learn all the names of cortical interneurons. Please try to know only their classification, the fact that they can be sometimes excitatory but usually inhibitory, and that it is possible to classify them more according to synapses forming with another cells. Next picture, there are also some pyramidal cells of the cerebral cortex, and next picture is showing special shapes of the interneurons, again just for your information, but I don't think that it is necessary to know it. Yes, and now let's look at the layers of the cortex. If you look at the slide in the left picture, which is stained with hematoxin eosin, you can see that there exists layers having more cells and having less cells. The layer which is really poor in cells is this one here, near the surface, under the meninges, and it is every time recognizable, and it is called lamina molecularis. Here we use word lamina, lamini. Cerebellar cortex had strata, but cerebral cortex contains 6 lamini. So lamina molecularis is the first, and it is recognizable every time because it contains really few cells. Then you can see a layer having more nuclei, many nuclei, from this, let's say, to this point. This layer having many nuclei should be so-called granular layer, lamina granularis. Lamina granularis, and as we are near the surface, it is lamina granularis externa. Okay, then we can see a layer having less cells. If we use higher magnification, we would find here more pyramidal cells, bigger pyramidal cells, and it is the first lamina pyramidalis, again externa. Then is usually very thin layer of many small cells. So it is the second lamina granularis interna. Then there should be a layer of the biggest pyramidal cells, lamina pyramidalis interna. And the last layer, which is at the border with white matter is here, is a lamina multiformis, containing variable number of usually big neurons. The right picture is impregnated, so here we can see mostly processes of neurons. Again, we can see some laminar arrangement of the cortex. The layers should be the same as we know from the left picture. Here are all the names written, and we know which cells are situated in which of them. Of course, it's much more complicated than is possible to study in the first year, but at least some information can be remembered. Lamina molecularis, it contains usually interneurons and inhibitory interneurons, and this picture is about the distribution of excitatory neurons. So we haven't got any excitatory neurons in lamina molecularis. Lamina granularis externa has got small pyramidal neurons, which can be sometimes oriented by their axon. Axon up, axon can go to the lamina molecularis, and there also can be some spiny stellate cells. The third layer, lamina pyramidalis externa, should contain middle-shaped pyramidal neurons or medium-sized pyramidal neurons having normal orientation, so their axon is going down and to the white matter. And also some interneurons, some inhibitory interneurons are here. The lamina granularis interna, it contains again some small pyramidal cells and also spiny stellate cells. And now we are finally in the lamina pyramidalis interna, where are really the biggest pyramidal cells of bats. Bat cells are here. And lamina multiformis, it's containing variable size of pyramidal cells, and again it is the third layer containing spiny stellate cells. So it is distribution of excitatory neurons in the cortex. If you look at this picture, you will see a distribution of inhibitory interneurons. I think nobody can remember this. Maybe you can remember interneurons of the molecular layer, Cajal-Retzius cells. They are quite well known, but then it is impossible to remember. It is just for information. All layers should contain some inhibitory interneurons. And here is a diagram showing their distribution, their quantitative distribution in all 6 layers. White matter of the cerebrum isn't so regularly oriented as was the white matter in spinal cord. You usually don't find parallelly organized axons, so there is irregular orientation of axons, and nuclei, which you can see belong to glial cells. Brain has got ventricles, and they are lined with ependym, the same ependym as you know from the spinal cord. Brain and spinal cord are covered by layers of connective tissue, which are called meninges. There are 3 layers of meninges, and you hopefully know them. They are called dura mater, arachnoid, and pia mater. Dura mater is a layer of dense connective tissue, which is firmly connected to arachnoid around the brain, but not around the spinal cord. There is, not to arachnoid, to periosteum. This is firmly connected to periosteum around the brain in the skull, but not in spinal cord. So in spinal cord there exists epidural space in this area, or not in spinal cord, in vertebral channel there is the epidural space. So you will see dura mater in the specimen of the spinal cord, but you will not see it on the surface of our brain specimens, because it is connected with the bone, with the periosteum, and so it's preserved in the skull during dissection. Next layer is arachnoid. It is tightly attached to the dura mater. This contact of arachnoid and dura mater is not anatomical. There aren't any anatomical junctions. There is just capillary force responsible for this contact and for its maintaining. Special flat cells are on both surfaces. They are on the inner surface of dura mater and outer surface of arachnoid, and these flat so-called borderline arachnoid and dural cells fuse together by this capillary force and hold the theoretical subdural space closed. Arachnoid is a layer of loose connective tissue, but atypical. This loose connective tissue of arachnoid is avascular. There are no vessels in the layer of arachnoid, and arachnoid is forming trabecules which connect it with next layer, with pia mater, and these trabecules go through the space which is called subarachnoid space, and which contains big vessels, or not big, normal vessels, which don't send capillaries, they don't send branches to the tissue of the arachnoid. The tissue of the arachnoid is supplied by nutrients, oxygen, and other substances from the cerebrospinal fluid, which is situated here in this space among the trabecules. So you already know the cerebrospinal fluid from the central canal and from the ventricles of the brain, and there is the second place where it exists. The system of ventricles is connected with subarachnoid space by several openings. You will know them from anatomy, and this subarachnoid space is responsible for the resorption of the cerebrospinal fluid into the arachnoid granulations. The last is pia mater, a layer of loose connective tissue covering the surface of a brain or spinal cord and accompanying vessels. So pia mater is going with vessels deep to the brain or spinal cord, and it is finished at the level of the last branching into the capillary network. Capillaries are not accompanied with loose connective tissue, but arterioles and on the other side venules, yes, they should be, or they must be accompanied by connective tissue. Okay, let's look at some pictures. At first, another schemas. There is arachnoid vilus and absorption of the cerebrospinal fluid from the subarachnoid space into the sinus. There are sinuses in the dura mater. Yeah, but this schema doesn't show anything new. And here is the first picture. Here is a brain. So the surface of our slide is not dura mater. Dura mater was connected with the skull, and so it isn't here present. So the first layer here is arachnoid, and we can see the border cells of arachnoid, which are organized like a simple squamous epithelium, but it isn't epithelium. It is a layer of special connective tissue cells, of specialized fibroblasts. There is a remaining tissue of arachnoid, and here we are in the subarachnoid space, where is a vessel going through, but this vessel is not branching and doesn't send any capillaries for the arachnoid proper. And here in the subarachnoid space there are thin trabecules composed of collagen fibers and some fibroblasts, forming connection between the proper arachnoid and pia mater. Pia mater is this thin layer of loose connective tissue on the surface of the brain, which is going inside together with vessels. So you'll find it also here around the vessel. If you have a specimen of a spinal cord, you can see the same situation, but also dura mater. This is superficial layer, dense connective tissue. It can be vascularized, dura mater, and here in this area you can see two layers of the border cells. So this nucleus or this nucleus or here these nuclei, they belong to dura mater. They are dural border cells, and when I use another color I can point I can point another border cells which are arachnoid border cells, and these are the cells touching themselves and forming a closure of the virtual subdural space. And here is a subarachnoid space. Oh, why? No. Yes. Here is a subarachnoid space. Oh, it can be blue. No, blue isn't good. Red is better visible. So subarachnoid space, vessel bridging this subarachnoid space, some trabecules, and pia mater on the surface of in this case of a spinal cord. So this is a layer of loose connective tissue where can be some capillaries. It can be normally vascularized, and it is well vascularized connective tissue. I think I see some erythrocytes in this location. So yes, or here surely. So pia mater is well vascularized layer of loose connective tissue on the surface. And here's everything in the electron microscopy. Border cells, dural border cells and arachnoid border cells and their contact, and you can never find any tight junctions or something like here. It is connected by a fluid situated in between, and a small piece of subarachnoid space and one trabecule containing trabecular cell, which is in fact fibroblast situated in this location. Pia mater is forming a special derivative in the area of brain ventricles, which is called choroid plexus, plexus choroideus. Choroid plexus is a bush-like structure entering all 4 brain ventricles. So it is situated in the roof of the 4th ventricle. It is found in the 3rd ventricle and in both lateral ventricles. The composition of choroid plexus is loose connective tissue of pia mater and superficial epithelium. You can see that it is simple cuboidal epithelium, which is in fact the derivative of ependyme, but in this case there is a connective tissue under the ependyme, not nerve tissue, and so in this case this ependyme develops the basement membrane and can be classified as a real epithelium and not only as epithelium-like cells or epithelioid cells, as in the case it lines the central canal or another portions of the ventricle and is in the direct contact with the nerve tissue. So epithelial cells are of the same origin. We will speak about the development of the nervous system, so you will know so-called ventricular layer of neural tube, and this ventricular layer can transform either to epithelium of choroid plexus or into the ependyme in another locations. Cuboidal cells of the choroid plexus are equipped with microvilli and also by kinocilia. Kinocilia can help move the cerebrospinal fluid, and microvilli are here important for the contact of cerebrospinal fluid, which is in the ventricle. So here is cerebrospinal fluid, and the function of choroid plexus is production of cerebrospinal fluid. So this microorgan is secreting or producing cerebrospinal fluid. It's difficult to speak about a real secretion because the fluid is mostly produced by transudation of fluid from vessels, and the cells of the epithelium are working more as transporters, more as the cells transporting water, ions and some low molecular organic substances from blood, than as real producers of cerebrospinal fluid. Cerebrospinal fluid should normally not contain many proteins. Proteins are not secreted by cells of cerebrospinal fluid, so it's more ion and water transport. to allow a good transport, capillaries of the cerebrospinal, capillaries of the choroid plexus are fenestrated. Here we can see some bigger vessels, but if you find real capillary, having really small lumen containing erythrocyte, one erythrocyte only, they are fenestrated. So these fenestrations allow the better transport of substances. So here is a detail of the fold of the choroid plexus, and it is one capillary, second capillary, endothelial cell, another nucleus of endothelial cell, and we know that these capillaries are fenestrated. It is impossible to recognize in the light microscope, of course. Epithelium of choroid plexus, as I said, some cilia are here, but mostly only microvilli. They don't only secrete or transport fluid from the capillaries, but they can also absorb something. They can also absorb some particles which are situated in the cerebrospinal fluid. And here is the electron micrograph, and in this case the cells of choroid plexus in this location have only microvilli on the surface. Many mitochondria, some smooth and rough endoplasmic reticulum, but no secretory granules are here, so they aren't like secretory cells. And now we are in the peripheral nervous system. So we are leaving CNS and its envelopes, and we can look at peripheral nerves. Peripheral nerves are composed of bundles of axons. So there are no bodies of neurons, they are just bundles of axons. These bundles of axons can be multiple, or the small nerve can be composed of only one bundle. If multiple, each bundle or fascicle of axons is surrounded by a layer, very special layer called perineurium, and the whole nerve is surrounded by epineurium, which penetrates among the bundles and fills all spaces among bundles of axons. Of course, the epineurium is well vascularized, arteries, veins penetrate to it, and capillaries can penetrate even through the perimetrium into the proper bundle of axons. Each axon can be surrounded by myelin sheath, but it isn't necessary. Some of them can be unmyelinated, but all of them must be surrounded by cytoplasm of Schwann cells. There is a rule which you know from general histology, but that there aren't any free axons in the peripheral nervous system. They can be free in CNS, in the neuropil, why not? But not in periphery. So each axon is surrounded by cytoplasm of the Schwann cell, which can form, which can form the myelin sheath. If we look at the bundle of axons in the higher magnification, we will see also the third new structure, which is called endoneurium. So there will be epineurium, perineurium, and endoneurium. Endoneurium is a material filling the spaces among the proper axons and Schwann cells and myelin sheath. It is mostly product of Schwann cells, but some fibroblasts can be here, also capillaries can be in the endoneurium, and the main extracellular component are reticular fibers here. This is a big nerve, cross-section of a big nerve composed of several axon bundles. Each of them is surrounded by perineurium. We will see a detail of perineurium, and remaining material found in the nerve is epineurium, containing, like in this case, vessels, bigger vessels. So epineurium is loose connective tissue, a little bit denser on the surface. There can be also adipose tissue. Perineurium is very special type of tissue. We will look at it. And endoneurium is here invisible because it is submicroscopic extracellular component of each of these fascicles or bundles. Now we are in higher magnification. Some blood vessel in the epineurium. Each bundle is surrounded by perineurium, and in this detail we can see axons, and most of them have got myelin sheaths. Myelin sheaths, in this case you can see it also in higher magnification. There wasn't so big extraction of myelin as we saw in the white matter of the spinal cord, and so the proper axons remained in the center of each sheath, and we can see some details of the sheath, for example these small lines here, which are called Schmidt-Lantermann clefts. And nuclei are here. Some of them can be fibroblasts of endoneurium, but more nuclei belong to Schwann cells. Schwann cells have got ovoid nuclei. Of course, their shape is, or their orientation is corresponding with axons, so we don't usually see ovoid sections in the cross sections of a nerve. In the cross section of a nerve we usually see small spherical sections of Schwann cell nuclei. If you use toluidine blue or another special myelin staining, Luxol blue also, or Weigert method, you can see myelin sheaths as dark circles. This toluidine blue has to be used together with osmium tetroxide fixation, but okay, it is not important at this moment. And it is also used here in this so-called semithin section. Myelin sheaths here are dark layers around the axons. Schwann cell nucleus is there. Endoneurium. Now in this magnification we can see some details of endoneurium. It is mostly network of reticular fibers containing some another extracellular components, glycosaminoglycans, proteoglycans, like in the connective tissue, but it is mostly product of Schwann cells. Schwann cells are special glial cells able to produce extracellular matrix. And it is the electron microscopy of a nerve, of a bundle of a nerve. Here we can finally see details of perineurium. Perineurium is composed of flat cells and some small amount of extracellular matrix. These flat cells resemble fibroblasts. So it's possible to say that it is a layer of special connective tissue on the surface, but they have another features which normally fibroblasts haven't got. For example, they have got superficial lamina like Schwann cells. And normally fibroblasts haven't got superficial lamina. They have got quite many contractile proteins like smooth muscle cells, and so they are able to contract a little and so protect the nerve bundle. They also have mutual contacts by tight junctions like epithelium. It is also special thing. And so perineural cells are really very special and unusual, and we aren't able to exactly classify them. The remaining parts of this picture show blood vessel. Not big, it is electron microscopy, probably it is a capillary. Continuous capillaries are in nerves. Then groups of unmyelinated axons. These axons are unmyelinated, and all of them are surrounded by a cytoplasm of a Schwann cell. So let's say this is one Schwann cell, and it nucleus is in another place. We don't see it, but it contains a lot of axons situated inside. The same situation is here, and the only myelinated axon is this one. You recognize electron-dense myelin sheath, and here is a piece of cytoplasm of a Schwann cell, and this Schwann cell was able to myelinate only one axon, but this axon is much bigger, much thicker than the others here in this location. Yes, this is another picture of myelinated axon. Now we can see also nucleus of Schwann cell in so-called peripheral collar or external collar of the myelinated axon. And mention collagen fibrils, which are building reticular fiber. It is a product of the Schwann cells and part of endoneurium. Unmyelinated axons. I think that you know these pictures from the general histology. Many axons encapsulated by a cytoplasm of a Schwann cell having its nucleus here in the center. Once more, the perineurium. Here is the picture of perineurium in light microscope. Layer of eosinophilic cells, nothing special. But if we... Sorry, I'll skip this and I'll maybe go back there. If we look at it in high magnification of electron microscopy, we can see superficial lamina on the surface of these cells, sometimes tight junctions between them, a lot of microfilaments resembling organization of thin myofilaments in a smooth muscle cell, also formation of pinocytotic vesicles, which we also know from smooth muscle cells. Here we know them as caveoli. So they have components and structures which are really not typical for connective tissue cells. The function of perineurium is to isolate the bundle of neurons against the electric impulses from surrounding bundles. So each bundle is isolated by its perineurium, and inside each axon is isolated either by a myelin sheath, is better isolation, or at least by the cytoplasm of a Schwann cell. I skipped the pictures showing longitudinal sections of nerves. You also know these pictures from general histology. If you use longitudinal section, you can see axon surrounded by myelin sheath, and this myelin sheath is from time to time interrupted by nodes of Ranvier nodes, which show the end of one internodium and the beginning of another internodium. Each internodium is made by the wrapping of membrane of one Schwann cell. And again, these lines crossing the myelin sheath, they are Schmidt-Lantermann clefts, where the layers of membrane are not so closely opposed, and some nutrition of the isolated axon, which is here in the center, is possible. The same here, note of Ranvier. I think that we can finish it. Another picture of perineurium, showing that there can be also reticular fibers among the cells of perineurium. So they have really properties of connective tissue, nerve tissue, muscle tissue, and if we take these tight junctions as the typical feature of epithelium, so also epithelial tissue. So all four tissues are in here in one layer. Epineurium, normal loose connective tissue, fibroblasts, collagen fibers, and other components of extracellular matrix. The last topic of my lecture are ganglia. Peripheral ganglia are the only places where neuronal bodies can be found in the periphery. So if you find, for example, only one neuronal body in a wall of some organ, you can be sure that it is a small ganglion. Peripheral ganglia are of two types. There are sensitive ganglia and autonomic or vegetative ganglia. Sensitive ganglia are so-called cerebrospinal ganglia. Cerebral ganglia are in the pathway of the cerebral nerves, if are sensitive, and spinal ganglia are situated in each root, dorsal root of the spinal cord nerves, and they are called spinal ganglia. This is the picture of the spinal ganglion from our collection, and let's look at its structure. This is a quite small magnification, but we can see even in this small magnification huge cells, huge, usually circular sections of cells, sometimes displaying a nucleus in the center, sometimes not. It depends on the level of the section. These cells are mostly accumulated in the periphery. You can see more cells, more cell bodies here around. Less bodies are in the center. Among the cells there are bundles of axons, and the structure is there similar as in the peripheral nerve. Another bundles are here around because the spinal ganglion is included in the course of the dorsal root, which has a structure of a peripheral nerve. These big cells here are pseudo-unipolar sensitive neurons. We can see them in higher magnification here. Their bodies are spherical because they send only one process. That's why they are pseudo-unipolar. And this one process is going from the body in one place. It is usually invisible. Where is it? And it's branching as a letter T. One is central branch, which goes to spinal cord or brain if it is the cerebral ganglion. The second goes to periphery, and it's called peripheral branch, central branch and peripheral branch. And this peripheral branch carries impulses from the peripheral sensitive endings. Both branches have a morphology of the axon. That means they are usually myelinated. So let's say that the whole process of our cell is the axon, and it is peripheral and central branch of the axon. Another thing which we can see in this picture is a variable size of these neurons. Some of them are really very big. They can measure more than 100 micrometers, but together with them there are smaller neurons. You must compare only neurons when you see a nucleus. So if you compare this neuron and this neuron, you can see their variable size. Of course, you cannot take into consideration sections without nucleus like this or this or this or this. We never can know if it isn't just tangential section of the surface of big neuron, so it is impossible. The variable size of neurons is typical for the spinal ganglion, and it's another thing according to which we can recognize it. Next thing which is here visible is a layer of cells, small cells, surrounding each of these neuronal bodies. These cells are satellite cells. They belong to glial cell family. It is the last glial cell, which we still didn't see, and they accompany the neuron, they protect it, they participate in its nutrition, similarly as astrocytes in the CNS. The layer of satellite cells here in the spinal ganglion is almost continuous because the neuron has got only one process, so it isn't interrupted by many processes like in the vegetative ganglion where neurons are multipolar. So pseudo-unipolar neurons, satellite cells, and of course their axons, or there can be also axons going through the ganglion, but usually there are axons of these our pseudo-unipolar neurons, either central or peripheral branches, and they can be myelinated in the ganglion. The whole ganglion has got a capsule which is continuous with the epineurium, and of course there is some connective tissue among the ganglionic neurons, which is also continuous with endoneurium. Okay, when we use semi-thin section and toluidine blue, we can see myelin sheaths well, and so it is the proof that the axons in the ganglion are usually myelinated. And here is a high magnification showing continuous layers of satellite cells around the bodies of neurons. This layer of satellite cell can sometimes resemble simple squamous epithelium, so be careful if you see the picture in the microscope. The last ganglion, or the second ganglion, is vegetative or autonomic ganglion. Vegetative or autonomic ganglia are of two types, sympathetic and parasympathetic. They differ mainly in location. Sympathetic ganglia are situated near the spinal cord in the truncus sympathicus, parasympathetic ganglia are situated more peripherally, and we can find them frequently in walls of organs as so-called intramural ganglia. And you already know them because we saw many intramural ganglia in the sections from GIT or in female reproductive system they were. And theoretically they can be in all inner organs which are innervated by a vegetative system. Vegetative ganglia contain less neurons. These neurons are more dispersed in the tissue. They are not accumulated either in periphery or central region. The neurons are in general smaller than neurons of the spinal ganglion, but their size is almost equal. There aren't so big extremes. Their size is around 30 to 50 micrometers, and there aren't any very big and very small neuronal bodies. Neurons of the vegetative ganglia are multipolar, so they have got more processes, more dendrites. Probably here we can see star-like shape of a neuron, stellate shape of the neural body, sending several processes. One of them is surely axon, the others must be dendrites. And that's why the body is only accompanied by several satellite cells, but the satellite cells never form continuous layer. There are some nuclei near the body, so they probably belong to satellite cells, but they never resemble any epithelium, as I said in the previous slide. Axons here are usually unmyelinated, so we don't find myelin sheaths here, and the cells situated in between the neuronal bodies are mostly Schwann cells, forming protecting envelopes around the unmyelinated axons. This slide, and it is the picture from our box, is probably from some very old persons. So the neurons contain brown pigment. You already know it. It is lipofuscin, and so you also know this picture because we have demonstrated lipofuscin in the picture of the vegetative ganglion, if you remember some time ago in the general cytology. Second picture is just higher magnification of the same. So neurons, sometimes with lipofuscin, only rare satellite cells on the surface of these neuronal bodies, never forming a big layer. Some Schwann cell nuclei among, and axons. Of course, also dendrites. There are also dendrites of these neurons receiving synaptic contacts from the preganglionic neurons from spinal cord or brain. The pictures impregnated, which can, better than hematoxin and eosin, give us a proof of the multipolar character of our neurons. There are many more than one process, surely many processes on the surface of the neurons. Just satellite cells and intramural ganglion. I think it can be enough, and we can finish our lecture. Thank you for attention. If you have any questions, you can prepare them for the practical training. Have a nice day and goodbye.